So we'll leave that up just for a minute while I introduce our next speaker. Dr. Ganapathy is from the North Campus, and he's the Associate Dean for Research for the College of Arts and Science. He's also Professor of Physics. And we've asked him to give a very brief, only 10-minute introduction about the resources that are available on North Campus. So I will hand it over to you.
Thank you, Karen. I'll just wait till the poll is closed, I guess.
I think you can go ahead and get started.
Okay. All good. Okay. Yeah, thank you. Good morning, everyone. And my name is Murthy. I'm an Associate Dean for Research at the College of Arts and Sciences. And I'm also, as Karen mentioned, I'm a professor in the physics department. So for this purpose today, I'll give you an overview of some of the core shared facilities on North Campus. And typically these fall under two kinds. Like one is actually the science and engineering. Okay, let me see. Yes, okay. Yeah, the science and engineering shared facilities. These are set up by the College of Arts and Sciences and the engineering school together. And those are managed by directors as well as facilities directors for each one of the facilities. The website is pretty up to date. And also you could Google it and then you will find this website. And also it has pretty comprehensive information on just move the poll to the side so that I could see. Right. And you could also see the contacts there. Don is the director for the instrumentation labs. And so there are two things I would like to point you in this slide is that you could also directly schedule lab times to use any one of the facilities. You could see this green button over here. And also you could search equipment, what would be useful for you in your research. So this is one cluster of shared facilities, which is the college and the School of Engineering put together. And the other one is actually maintained by the chemistry department exclusively under the College of Arts and Sciences. And that has nuclear magnetic resonance facilities, as well as mass spectrometry and so on. I'll show that at the end of these 10 minutes, okay? Okay, and if you click on those links that I showed in the previous one, the search equipment thing, that'll take you to this searchable index that you have over here. You could search by function, like whatever your interest is, chromatography or microscopy that you would like to use. Or you could also type by location or the facility type and so on. And you could see the left column here that tells you some of those categories that actually are coupled together to show you what kind of facilities are available on North Campus. I'm showing all of these that are on North Campus. And this list has everything except what's recently added to these facilities cluster, which is the digital manufacturing lab. I'll show you a separate slide on that in a moment, because there are many people in the health sciences, they start to use these digital manufacturing lab for three-dimensional, 3D objects, 3D printing, as well as small parts and so on. Okay, if you click on any one of this, like if you click on any one of these browse by function, that will take you to a very exclusive list of things. I won't go through all of them one by one, but you get an idea of what kind of facilities that are available. And again, each one of those is the link that you can click, and you'll get more technical details of those microscopy as well as tissue culture or cell culture facilities that you would like to use. And one thing that I would say about the microscopy is that it's a very comprehensive facilities that we have on North Campus. I'm sure that there are similar or like comparable kind of opportunities available both on the south as well as on the downtown campus. What the North Campus has, in addition to those, is actually an atomic force microscope that if you're interested in using, as well as some of the scanning electron microscope that is attached to the clean room for like lithography and things, if that's of interest to your research mission. And of course, Raman spectroscopy, as well as microscope and optical microscopes that are pretty much standard ones that you would get in many of the facilities. And right. And on the same page from the top one that I showed you, you could also click on the rates. That tells you what's the current rate. You could also have your students or postdoc getting trained in, have an initial orientation as well as training through each one of those facilities. There is a very generic training and that everyone has to go through for safety and everything. And after that, if there is specific instruments that you would need to have trained, you could also set that up. And also the staff that are available in this facility, they would offer you up to 3 hours of technical support to your project with no additional cost once you sign up for training and everything. Okay. And after that, depending on your project needs, you could also have project consultation and processing and so on done with the help of the staff, or you could do it on your own. Okay. And the digital manufacturing lab has become more popular recently. This is a recent addition, probably a few years ago. And there are many faculty members at UB who now use these for 3D printing and additive manufacturing kind of technologies. And they have the software as well as I think there are about 4 equipments there that actually will help you depends on your needs, what kind of material or what kind of precision you need in their machining and everything. They could do these things too. And on the same page, you would also see the getting started link. And that again, that they have, at least for the few of them, there is a new user guide. You click on them and then it'll tell you what to do, actually whom to contact and how to set up time and so on, as well as where they are even located, right? North Campus looks like a zoo to many people from the other campuses. And so it kind of tells you what buildings these are located. And the biodesign core facility is in the Bonner Hall, and the materials characterization lab is in the furnace hall, and the clean room is in the electrical engineering building, which is the Davis Hall. And the TEM facility that you see here, the high resolution TEM, that's being replaced currently. There is a new one that's a $2 million TEM that's just recently being purchased. It should be here sometime during this semester, and the installation is kind of scheduled in a few months from now. Okay, and that's one set of equipments and shared facilities that you could access on North Campus, which is the science and engineering cluster. The other one is mainly maintained by the College of Arts and Sciences, and these are all located in the Natural Sciences Complex under the Chemistry Department. Chemistry Department operates it. The college oversees this. There are 2 main things that I would want to point here. One is that this is the very large state-of-the-art NMR center, magnetic resonance center, and you could see at the bottom of this page what kind of NMR. There are a couple of Bruckers and there are a couple of variants, and also they have an EPR spectrometer that it's again managed by one of the facility director, and there are 2 scientists who actually take care of operating this machine. So you're welcome to. They have their own website to actually tell you about the facilities that are available there. And the second thing is they have a large set of mass spectrometer and chromatography setup too. You would also have seen that shared facilities that I showed earlier has mass spectrometry capabilities because chemistry has a very large volume of mass spectrometry they do. So there are 2 clusters. These are mainly used by chemistry, but it's a UB facility. Everybody could use this, and they're very heavily used. And with that, I'll stop here. These are all the North Campus. I guess there are people queued up to talk more about the South as well as the downtown campus facilities. If there are any questions, feel free to, and on the website, the facilities director's contacts for each one of them are listed, as well as any questions about North Campus facilities, feel free to email me. I'll be happy to talk. Right. And thank you, Karen, and thank you, everyone.
Thank you so much for that. So now, to stay on time, I'm going to quickly go over to Dr. Halterman, who's going to talk about the resources downtown. Dr. Halterman is the new Senior Associate Dean for Research at the Jacobs School of Medicine and Biomedical Sciences, and also the Executive Director of the Office of Research. Dr. Halterman?
Good morning. Thanks, and welcome to newcomers to the institution. I'm probably wearing out that badge. I've been here for almost 10 months, so I think once October rolls around, I'm inculcated in UB culture by that point. Delighted to talk about cores on the downtown campus. Let me pick a share here and see if we can get this loaded up. So pleasure to give this talk. As Karen mentioned, I'm the Senior Associate Dean for Research at the School of Medicine, a faculty member in the Department of Neurology, and have a laboratory in the JSMBS. I also run the MD PhD program. So this is a great opportunity to learn about some of the interesting background of Buffalo. So a little history lesson through real estate. The Buffalo Niagara Medical Campus is really a force in evolution, and If you look back to its roots in 1898, the Roswell Park Cancer Center was initiated, and its complex has continued to grow since that time. Let's fast forward a little bit to the 2005-2006 era. So Hauptmann Woodward's built their complex, and CVLS went up in 2006. And interestingly, CTRC in 2012. So keep in mind that the hospitals had been here for quite a long time. Children's Hospital in a prior location before Oshai, and Buffalo General. And until 2012, there hadn't been much of a research presence on campus. And so the building, the 23 marked there on the map, this was really an important shift in the landscape, providing an opportunity for translational and basic research to live very close to the hospitals. And then, of course, in December of 2017, the JSNBS opens its doors. So we are a confederation of research enterprises and collaborators and colleagues. So I'm going to cover cores that are across each of these different areas. Within the JSNBS and in cooperation with the cores in South Campus, Pathology and Anatomical Sciences have a research service center that's directed by Wade Sigurdson. That center has a number of attributes: histology core facility, confocal microscopes, and flow cytometry. We have multispectral imaging capabilities, EM, as well as imaging mass cytometry, which was added most recently, and I'll talk about these in course. So the histology core facility, not surprising that this is linked with pathology, right? Services include many of the things you would expect: paraffin embedding, frozen histology, typical staining, IHC, in situ, et cetera. And the instrumentation is what one would expect for this type of a high volume center. The confocal microscope and flow cytometry facility, constantly in evolution, bringing on new equipment. We have a number of Leica instruments for confocal, and we are updating our flow cytometry, most recently acquiring a Cytec Aurora, which provides a number of advantages for investigators, increasing the number of antibodies that we can use in our analyses at one time. And then other systems, the Celeste from BD, as well as an area fusion sorter. We host a range of equipment for molecular biology. This is a partial list, and these are used frequently. The multispectral imaging suite, this may not ring in your ear, but let me tell you a little bit about it. The idea is that we're able to capture across not just visible, but taking advantage of UV and infrared bands. And so you end up with high resolution, multidimensional view of tissue samples, and the signal to noise is improved. So this has a lot of applications for label-free imaging of tissue. These services include whole slide scanning, digital annotation, and of course, there's training and consultation for students and faculty. The EM core provides services including sample preparation, embedding and labeling, standard TEM, and allows for ultrastructural analysis of tissues and cells. And these systems, if you don't use them, just to point out, we're talking about magnifications up to 600,000x. So we certainly get into the details of our science here in this core. The imaging mass cytometry I'm very excited about. So again, on the throughput side, this allows you to utilize antibodies that are conjugated not to fluorophores, but rather to stable non-radioactive metals, oftentimes lanthanides. So you can batch stain in a tissue specimen 40 plus markers, and there's no autofluorescence. And this is particularly problematic in tissues like lung, for example, where you have a lot of birefringence and background. You can co-detect protein and RNA. So this is backed primarily by a Hyperion XTI, and it has CyTOF capabilities. So this system allows interrogation of both tissue as well as single cell analyses using the same antibody panels. The other resources, I add toxicology research, which is on South Campus, but it's relevant to many of the investigators here, and I don't know if it's going to be covered elsewhere. So this is run by Paul Kostiniak. Analytical toxicology, performing things like ADME kinetics, antimicrobial testing, and toxicogenomic assays. So for any small molecule developers, drug target developers, this is an important core to know about. On the translational and clinical side, the Institute for Healthcare Informatics is a very important resource that's been around for over a decade. It's a data repository of electronic medical records and claims data for Western New York. I believe they have over 1 million patient encounters in place, data access, secure storage, computing, and analytics for these kinds of data. And the expertise is broad and absolutely required. So making sure that these data are secure and adhere to HIPAA standards. There's expertise in human subjects research, using tools to be able to interrogate that data like I2B2. And then they have facility using Trinetics, which is clinical data that is warehoused from industry trials, which can be very useful in thinking about prospectively considering trials in a certain disease area. I came in just as I was hearing about the MRI scanners on North Campus. We also have imaging here at the CTRC building. Last year acquired a new Philips MR7700. This is a 3 Tesla that can be used for both humans and preclinical imaging, as well as a preclinical scanner from Brucker. We have investigators across multiple departments with broad expertise in quantitative image analyses, and I would recommend that you speak with Dr. Schweitzer if you have questions about those. And then the Neuropsychology Center. So it's one thing to have patient data. It's another thing to have patient imaging data. But what really brings this together is if you can link that with some of the functional outcomes. So development and implementation of cognitive outcome measures, psychometric testing for things like personality traits, psychiatric symptoms, cognitive functions, et cetera. This is really the sweet spot where Dr. Benedict shines. So for those of you that are interested in translational neuroscience, these 3 resources are useful. So I'd be remiss if I also didn't highlight the UB Biorepository. This is housed on the 6th floor at the CTRC. This is led by Dr. Norma Nowak and John Tomaszewski, and then Don Yergau as Associate Director, and Trevor really runs the show there. So if you haven't contemplated what a biorepository is or what it can do for you, at its essence, it is a highly standardized, secure way to freeze, store, whatever you'd like to call it, preclinical and clinical specimens. This allows investigators to remain competitive, allows you to stretch your research dollars, and it eliminates duplication in labs. So we all have our -80s. We all know that there are things in there we haven't looked at for quite some time, and we know that some of those contents are indispensable. So the biorepository is really intended to safeguard our research resources, but more than that, develop collaborations and think about ways in which we can really expand the science that we're doing. So the processing services ensure data quality and regulatory compliance. So just looking at some of the areas in which investments have been made in sample collection and processing, quality control analysis, the storage facility itself, and workflow automation, I'll just make a couple of highlights. So of course, they have systems to be able to do sample processing and dispensing. What you are dispensing, you want to make sure you're putting in the tube and freezing and paying for on a monthly basis as of high quality. So the control analyses are really vital in this area. And then the capacity is quite astounding. Right now, we've got about 120,000 biosamples stored with a capacity of near 1 million. So take advantage of this resource. If you have questions, please reach out to Biorepository at Buffalo EDU and learn more about what potentially this could do. And certainly think about writing this into your proposals. Begin with the end in mind. If you anticipate needing storage, long-term storage needs, this sort of forethought is considered really important, and reviewers will look at that with, I think they'd be impressed. So what else? Center of Excellence in Bioinformatics and Life Sciences, CVLS, hosts the UB Genomics and Bioinformatics Core. For many of you, these are probably resources you're already currently using. In addition to some of the standard genomics approaches that you can see on the left, we also offer bioinformatics services run by Jonathan Bard. So it's one thing, again, to have data in hand. It's quite another to have help and support in being able to analyze very deeply the data that you have and not leave anything on the table. So I'll leave this, and you can certainly refer to the list going forward and consider its utility. CVLS also has a few additional resources. There's a mass spectroscopy core, the Center for Computational Research, and there are several ISO certified clean rooms for microfabrication, photolithography, and characterizing materials. If UB does anything, we do biomedical engineering and engineering exceedingly well, and these are just a few of the clean rooms on campus, but we're delighted that they're here down on the VNMC. Hauptmann Woodward has a number of resources, cryo-EM, they have a National Crystallization Center, as well as synchrotron services. Edward Snell is the contact to reach out to them for further questions. And I'd be remiss if I didn't point out the excellent cores that are managed by Roswell Park. Many of these fill some gaps in our offerings. Drug discovery, there's a gene targeting and transgenics core, excellence in nicotine and tobacco product assessments, and of course, expertise in translational immuno-oncology. They have had a number of resource days, and if you have not been able to attend, keep an eye out. It's worth your while to understand what they have on deck there. We work very closely together and appreciate very much their support, particularly at times when we have equipment that's being serviced and we don't want our investigators to be slowed down. So I will stop there and happy to take any questions, but hopefully that was informative. And if you have questions for me, don't hesitate to reach out.
Thank you, Dr. Halterman. Actually, you were just mentioning about the resources. We do have an upcoming resource day that will include a lot of the cores and facilities available at downtown, including the Roswell Park Comprehensive Cancer Center and the Jacobs School. So keep your eyes open for announcements for that coming up soon. To stay on time, I'd like to quickly pass this over to Dr. Keith Kirkwood, who's a Senior Associate Dean for Research for the School of Dental Medicine on South Campus, and he will talk about the resources there. Dr. Kellerman, or Dr. Kirkwood, I'm sorry.
Hey, good morning, everyone. So my name is Keith Kirkwood. Can I share my screen, Karen? So I first thank Karen and Jill for organizing this. I think it's incredibly important to show and highlight resources. I'm going to highlight some that are at the dental school here on South Campus. I'll share my slides now. Can everyone see these?
Yep.
Okay. So there are certainly going to be other resources here at the South Campus. I'm just going to highlight 3 cores that are really located here in the South Campus, specifically in the BRB. So the dental school, for those who are mainly on the South or downtown campus, the South Campus has the dental school, and we've been a leader in oral health research for decades. We have been consistently ranked very highly. We're smaller. We only have somewhere around 40 full-time faculty. We've increased some, so I'm not quite sure what the number is right now. But we have, really just a fraction of the faculty that the school, the Jacobs School, has. And we've been consistently ranked in the top 10 for many years for research. Currently, we have 3 training grants. We have 2 that are active right now. One is a no-cost extension. And so we're very fortunate to have a lot of training opportunities for our students and trainees here. And so the Shanghai rankings have us at number 7 in the US and 11th worldwide. So we're very proud of that. So with that, basically, probably similar to what Marc was sharing with you, we have a website that has all of this information. So if you don't know, just go on the UB website, look for the SDM, and within that, we have research as one of the tabs. So all of the information I'm sharing with you today can be found there. We have 3 core facilities here located in the Biomedical Research Building. And these are going to be the materials testing facility. I don't think that I wasn't here for the North Campus presentation. I'm not sure if those facilities exist within engineering. And then we have a materials analysis core and an optical imaging and analysis facility. Some of these services are going to be redundant with those offered downtown, but I want to certainly highlight what we have here that is going to be different. So the first facility is a materials testing facility. This is overseen by Sabrina Sahaki, is a new recruit who is a dentist scientist, and she has a facility director that she works with, Steve Michalka, who is a materials scientist. We are actively providing a short list of services here. There's a lot more that they can provide. We have a number of equipment dedicated to materials testing, Instroms, other instruments that are available. These are available, and we're really in the final stages of getting chargebacks approved by the North Campus so that we can offer these services to everyone. But they're fairly unique in terms of what materials testing can be offered. Most of these services are with calibrated instruments that adhere to ISO standards. So many companies are going to seek out these opportunities to do materials testing in the region as well as throughout the US. So again, Steve is the director for this facility, and Dr. Sahaki is the faculty director for this facility, and there's a website there. And this facility is located on the 3rd floor of the Biomedical Research Building. The next facility is Materials Analysis Core. This used to be called the South Campus Instrumentation Facility. Peter Bush, shown here, has been doing this for over 30 years. He contracts with a lot of companies outside of UB, but he also has training for people looking at SEM, X-ray diffraction, Raman spectrometry, other opportunities for looking at failure analysis. And he does a lot of consulting with individuals and provides a lot of services to UB as well as the region in general. His core facility is located on the 4th floor of the BRB. And again, there's a phone number and website to contact Peter. The last shared facility that we have, core facility, is run by Andrew McCall, and it's entitled the Optical Imaging and Analysis Facility. This is also located on the 4th floor. We have a number of imaging microscopes, including an Andor Dragonfly spinning confocal. We have an axial observer, inverted fluorescent, and an upright axial imager fluorescent microscopy. We also have a Fortessa cell flow sorter analyzer. Marc also mentioned that they're getting a new spectral flow cytometer. We are as well, because the Fortessa is probably reaching its life expectancy at this point. And so we're pursuing with getting a spectral flow cytometer. We've also been able to secure funding from the VPR's office in the last couple of years to obtain a flow sorter. So we have this available. One unique resource that we have here is a Scanco 100 computed or micro CT computed tomography. This is not available anywhere else on campus, as far as I know. There is a PET CT available in the CTRC, which does larger samples. This is for micro CT. So as you can imagine, we can use this for mouse samples as well as rat, and we can go down to as low as 4 micron splines that we can scan. We also have an in vivo imaging analysis machine to look at tumor growth in vivo. And then we also have a small animal irradiator. This is a non-cesium. This is an X-ray source irradiator that we can use for bone marrow chimeras. We use it for also inducing oral mucositis in some models. This again is run by Andrew McCall, and he is located on the 4th floor of the BRB. So I'll stop there and take any questions. I'm getting everyone back on time.
Thank you so much, Dr. Kirkwood. Before we take a break, we would like to allow Kyle Kellerman to talk about the Health Sciences Scholarly Project. So I will let him present, and I've also sent out, if you'll see in the chat, there's a link for a survey that he asked to distribute. Are you ready, Kyle?
Absolutely. Thank you so much.
Hi.
Hello, everybody. Thank you to Jill and Karen and everyone else for setting this day up today. It's been fantastic so far, and I look forward to the rest of it. As she said, my name is Kyle Kelleran. I'm an assistant professor in the Department of Emergency Medicine, and I'm also the director of the Health Sciences Scholarly Project in the new Well Beyond curriculum. I've been granted a few minutes today to introduce the Health Sciences Scholarly Project to all of you. Some of you may know, and some of you may be aware of this new requirement. Some may be new to you, but as part of the new curriculum, all of the students will be required to complete a new research project. So they're going to be required through their time to complete this research project. It is part of the scientific literacy and inquiry pillar, and that has been designed to teach medical students to read, understand, use, and produce the scientific literature and knowledge. And again, this project, the Health Sciences Scholarly Project, is kind of the application of that learning. So again, with these new first-year students, medical students, they will be required to complete this research project as part of their graduation requirement, and all of the future classes, after this first year will as well. We've designed this to accept a wide array of different types of projects across many domains, from bench science to clinical to bioinformatics, business, and beyond that. All the deliverables for this project have been kept to a minimum, and they're all designed to enhance our students' resumes and marketability in preparation for applying to residencies, as well as actually preparing them for their residency interviews. A couple of examples of this requirement are they need to present this research at our research forum. That would add a line to the resume, of course. We're also requiring that they develop an elevator pitch about their research for use in the residency interviews. I've seen it numerous times where students haven't been able to discuss or talk about their projects in depth. So the people who are interviewing them may not know what their depth of knowledge and involvement actually were. So we want to kind of work on some of those things as well. Again, with this new requirement, we will need to start to develop and secure approximately 100 to 120 new mentors and projects per year. So that's a pretty big lift, but we need to accommodate 180 students, around 180 students per year, every year, ongoing, moving forward. Over the coming months, we'll be reaching out to the department chairs, research directors, other stakeholders on campus, trying to line up projects and mentors for our students. The department chairs were kind of introduced to some of this at, I believe, the August chairs meeting, if I'm not mistaken. But we're going to start reaching out to everybody starting here and moving on forward, and trying to develop some relationships, build some projects, ideas, and eventually get a database together of information for the students as well as the mentors. I did want to emphasize that this project for the students is about mentorship, learning, and experiencing the research process. They do need to fully understand the what's, the whys, the hows about the projects that they're working on. So it's not just, let them pull some data. They do need to understand a lot more than that, a lot more in-depth involvement than just that. We will be working to pair our students with mentors starting in January of their first year, so that will be coming up faster than we all expect. And then they will have about until January of their fourth year to complete that project. Some of the students will complete their projects early, others will take the full time, but as a mentor, you would be able to determine the best way to guide your students or student through this process with support from our team. Students can be on research projects as individuals or small groups of 2 to 4 students. We're going to kind of limit it to a max of 4 students. And because this is an ongoing program, you would also have the opportunity to bring students on in a legacy format. For example, taking 1 student each year and keeping them on projects long term. So you can kind of roll one to the next. They can kind of keep training the next generation and keep things going on. I know there are a number of projects on campus that are continually collecting data and analyzing and moving things forward. I'm really excited about this project. I was really excited to get involved with this because I think it's a great opportunity for our school to bring us as researchers together in a singular mission, kind of guide the next generation of clinicians, and hopefully maybe draw some more of our faculty members into the research world. And of course, with an end goal of hopefully increasing the productivity of our school as a whole. I have a flyer I will be sending out. You'll probably be seeing a few flyers sent out from HSSP. There's also, obviously, Karen said, the survey link in the chat. If you wouldn't mind filling that out for me, I really appreciate that. If you ever have any questions, comments, concerns, feel free to reach out to me. We have an email address set up that goes directly to me and my team. It's hssp@buffalo.edu. And other than that, I want to thank you for your time, and I hope you enjoy the rest of your day. I won't hold you back anymore from our short break here.
Thank you very much. It sounds like an excellent program.
Thank you.
So yeah, we'll take a quick break and then we will start back up at 10:30. So I'll see you all soon.